Good morning, everyone. My name is Charles Geddes. I'm a research scientist with Agriculture and Agri-Food Canada, based out of Lethbridge, Alberta. And today I'm going to be talking about genetic testing for herbicide resistance. So first of all, herbicide resistance is a growing problem across the prairies. The majority of farmers are dealing with at least one type of herbicide resistance. And traditionally, the way that resistance was confirmed was you'd have to let the weed go to seed. You would have to collect seed from that population, send that into a lab. They'd grow it up under a controlled environment, treat it with the herbicide, and then look at the phenotypic response to see whether it responded like it should to the herbicide compared with resistant and susceptible control populations. What we're doing differently here is we're trying to substantially shorten the time frame that it takes to confirm resistance, and the way we're doing that is through molecular biology. So what we're doing is we study herbicide-resistant weeds, we confirm what is the molecular mechanism that confers resistance in those populations, and then we develop rapid tests that can be done in a lab to confirm resistance, just using DNA that's extracted from leaf tissue. So we just finished a project that was funded by Ardar over the last 4 years to develop genetic tests for herbicide resistance, and we've developed about 14 genetic tests. And one that we'll be talking about today is looking at group 14 resistance in kochia. So we designed this kit here, and the kit is really simple to use. It's a sampling kit. What you do, first of all, it just comes with a set of instructions, and it's a larger Ziploc bag, and inside that are 10 smaller Ziploc bags. And what we're doing is we're sampling leaf tissue from kochia and putting it in envelopes that go inside those smaller Ziploc bags. So we're sampling 10 plants per kit, one kit per field, and that's essentially it. So we can look at the kit here. So we have our instructions that come with it, and then there's 10 of these smaller Ziploc bags with just information to fill out there for the weed population that you're interested in. These orange beads in here, these are just silica gel beads, and that's used to desiccate the plant material once you put it in the envelope and then the envelope in the bag. So to use it, all you do is you just open it up, grab your envelope here, and let's go sample some kochia. So it works best on a weed population that's been treated with the herbicide that you're suspecting it is resistant to. So in this case, group 14 resistant kochia, for example, in a field that's been treated with group 14 herbicide, and we're seeing that kochia isn't controlled. So what we're looking to do is we're just sampling some of the newest growth on the plant. So we want nice, lush green tissue here. So we'll choose this one. We're just going to snip off here about a 2-inch portion. We want to make sure there's about 4 to 6 leaves on there. We place that in our envelope and close the envelope up here and put it back in the Ziploc bag. You seal that up, and over about 24 hours, as long as the tissue is not really wet, that tissue will desiccate and it will preserve it until you can send it back to a lab. In the lab, we just take the tissue, extract DNA, and use our assay that we designed to look for the mechanism that confers group 14 resistance in kochia. So knowing what type of cross resistance that mechanism confers will tell us what herbicides will work and which ones won't. Simple as that.
Is there any limitations on the stage of the weed or the size of the weed that you would sample?
So it works best to use the actively growing green tissue from a plant, right? So if your plant is really desiccated or it's being controlled well by the herbicide, that just means that it will be a little bit more difficult to extract DNA from it, right? So it's just about the yield of the DNA that we can extract, and it works best on nice, lush green actively growing tissue.
And it's only for group 14, not 249 or anything. That's a whole separate. Like, why is it just 14?
So right now we're just doing group 14 resistance because this is a new issue that's showing up on the Canadian prairies, and the idea is that we're wanting to get some information on how widespread is that issue. So we know that group 14 resistance in kochia has been confirmed in Saskatchewan, Alberta, and North Dakota. We're hearing about some potential cases in South Dakota as well. And so far, we've only confirmed it in 2 fields, one in Alberta and one in Saskatchewan, but we do suspect that it is quite a bit more widespread than that. And so um, using this sampling, it lets us know how widespread is the issue, but it also lets the farmer know what herbicides will continue to work on their population as well.
So then, for last question, for are you looking like, so you're doing sampling for the group 14 resistance, trying to understand, are you looking for farmers to volunteer to kind of help you get the sampling, or do you have a list that you're kind of set up? Like, how are you trying to get, I guess, producers engaged in this process?
Yeah, that's an excellent question. So essentially, what we're doing this growing season is we have a pilot project where there's an open call for if farmers or agronomists want to submit samples. We can send a kit out to them. They collect the samples and send it back to our lab, and we'll do the lab work and provide them with the result. So there's an open call this summer. If somebody wants a kit, they're welcome to contact me, and we can move forward with shipping them a kit, and they just sample, then ship it back to us.